Good afternoon, everyone, and welcome to this week's Institute for Quantitative Biomedicine seminar. I'm Stephen Burley, the founding director of the Institute, and I'm delighted that our colleague Darren York has agreed to invite and host another interesting speaker. So I just want to draw your attention to the fact that the series is nearing the end. On the 29th of November, we'll be welcoming Jeff Gray from Johns Hopkins to Rutgers, and he's going to be talking about AI tools for antibody engineering and protein docking. He's part of the Rosetta Mafia, and definitely somebody worth seeing in action if you're interested in protein design, antibody therapeutics, etc. And then our former tenant, Renew CO2 Incorporated CEO Anders Larsen, who's gone on to greener pastures with money from the Gates Foundation to support his bid to convert CO2 into fuel, will be coming and giving us a progress report to acknowledge the contributions that the Institute made in helping him and his team get started here at Rutgers. They've now gone on to a commercial real estate space in Somerville, I think it is, spending literally millions of dollars a year to accelerate the development of their product or products. So with that, I'm very pleased to introduce Darren York, who will introduce today's speaker. Darren, thank you.
Hey everyone, and thank you for coming. I am absolutely delighted to have here with us today Aaron Robart from West Virginia University. Aaron does a variety of things. He's, of course, highly recognized for his research contributions, but also is very active in the educational space. His overarching goal is to unravel the structure, function, and mechanisms of catalytic nucleic acids, which is also, from a computational standpoint, what my group works on. And so we're extraordinarily excited to have him here with us. He got his PhD at University of Calgary in Canada, and then went on to do postdoctoral research at UC Berkeley, and is renowned in the field for looking at things through both biochemical studies and also structural biology studies, working in the splicing field. And today, we're delighted to have him here with us. I should also mention he has some recent awards. He had the Powell Junior Faculty Research Award and West Virginia University Provost Research Award for COVID assay development. He has a number of active grants and is just extremely active in a lot of areas. And in particular, from our standpoint, very excited to hear about his pioneering work in the understanding of catalytic nucleic acids. So without further ado, I'll hand you over to Aaron for his talk.
Awesome. Thank you for the kind introduction, Darren. And thank you for the invitation to come out and see all the really wonderful science out here. I've had a fantastic morning. I always say I need another project like I need a hole in the head, but I think I've got about 5 new projects and I'm already really excited about just with the conversations this morning. Okay, so like I said, I'm in West Virginia University in the heart of Appalachia, trying to do structural biology. So I really always like to start off from a historical perspective, and this is actually one of my favorite slides. So David Phillips at the Royal Society of London is presenting the structure of lysozyme. And what David has done here is he's taken his 3D structure that he solved by crystallography and placed it on the lectern beside him, and then suspended from the ceiling of the auditorium, dangling pretty much to the floor, is the primary amino acid sequence at the same scale. Okay, so at the time, people looked at this and marveled that a protein could somehow go from this long dangly string of amino acids and fold up into a complicated 3D shape that can actually accomplish chemistry. However, maybe not in this room, but most rooms I go into, when we talk about things like RNA, when we talk about things like DNA, I almost guarantee that you have this image of a long, unstructured, dangly, single-stranded moiety, as opposed to something that's actually folded up into a functional shape. And if I accomplish absolutely nothing else today, but to have you start thinking about nucleic acids as having a structure that can do interesting chemistry, then it was a trip well taken. Okay, so I actually started off as a political scientist when I went to university. And I thought I was going to be a lawyer, but I was still taking chemistry courses. And I remember I had one particular lecture where they were talking about this RNA world, that before there were cells, before there were proteins, DNA, there was a primordial soup of RNA folding into shapes and figuring out the chemistry of life. Needless to say, that had a tremendous impact on the course of my career, and I switched into pretty much studying them for the rest of my life. So we like to think that RNA goes far beyond just the roles of core components of the central dogma, mRNA, rRNA, tRNA, and that we can think about the RNA world hypothesis as being something that really formed fundamental machineries and processes that we still see as molecular artifacts in modern day cells. So evidence for this would be, say, things like the ribosome, highly structured RNA that actually promotes the peptide bond formation. Another great example is things like the spliceosome, 5 different snRNAs coming in on a very complicated dance to remove introns and make mature messages that we use to encode proteins to do very important cellular work. Now, understanding the function of the spliceosome and its humble origins, likely from ribozymes that we found enriched in bacteria, is the bread and butter of research in my lab. But I want to spend today talking about new directions that we're doing, thinking about how DNA can also act as an enzyme. Okay. So the concept of a catalytic nucleic acid is fairly well cemented at this point. In the early 80s, Tom Cech found group 1 introns and its role that it was playing in ribosome maturation, tRNAs, in particular tRNA maturation through RNase P. And these ribozymes can come in a variety of sizes, different flavors, okay? Very small, like the hammerhead ribosome, one of the first ribozymes that we had a structure of and could understand its mechanisms, all the way up to the very large ribozymes that my group studies with these highly structured group 2 introns that do a splicing chemistry. But in the early 90s, Ron Breaker, Gerald Joyce asked a really interesting yet very simple question. If RNA can fold up and be catalytic, why not DNA? Is this something that DNA could also accomplish? So they set out to test that hypothesis that maybe DNA could also encode some sort of fold that would allow it to have chemical action. And to accomplish this, what they did is took a folded oligonucleotide with a single ribose position, 40 randomized positions, and a biotin label. We could then attach this randomized pool onto a streptavidin column, and then screen through addition of different cofactors, in this case magnesium, to ask, can we get a DNA-dependent or DNA-catalyzed site-specific RNA cleavage reaction? Which we'd see by basically the winners of the randomized pool eluting off the column. We could then take these winners, do PCR to reamplify, regenerating the cleavage site, putting the biotin site back on, and go through multiple rounds of selection and enrichment. So they did this and came up with the first catalytic DNA, or a DNA zyme, which was called the 1023. 10 meaning I did 10 rounds of selection, 23, which means I was clone 23 and judged to be biochemically the winner of the contest. Okay, so over the last several years, my group has been really focused on trying to get structural insights into how the 1023 DNA enzyme can actually promote a site-specific RNA cleavage. We really honed in on this DNA enzyme for two reasons. One, it's just historically very important. It was the first one discovered. People have been craving this information for 30 some odd years. The second reason is that it can identify and site-specifically cleave RNA with high dependence on a magnesium cofactor, which means that this might actually be something that you could use in living cells as a therapeutic agent. Okay, so let's spend a couple seconds and just look at the nuts and bolts of this DNA enzyme. The first thing you might notice is that it has two flanking arms with just denoted N, which means I'm random. What this implies is that you can have a core catalytic component, which is this loop here, and attach it on to reprogrammable arms, which means I can direct this RNA cutting DNA machine to bind very specifically to almost any RNA of interest. Once bound, it catalyzes the cleavage at only one unique site located between an R, meaning I'm a purine, or a Y, a pyrimidine. So you get all your specificity directing from the binding arms, and then you actually have a fairly malleable, in a sense, because you do have choices, position where your DNA-directed scissors can cleave and open up the RNA. One other really interesting thing about DNA enzymes is that after they promote this DNA-dependent RNA cleavage, they can release and turn over, allowing them to bind another copy, cleave, turn over, which make them very powerful as a potential knockdown agent. You don't bind, cleave, and you're stuck. You can turn over. Interestingly, so far, these DNA enzymes have only been involved in a test tube. We've never found a naturally catalytic DNA, and maybe that makes sense. We trust DNA to be the guardians of our genetic information. We probably wouldn't love it if it started to have rogue activities going off and functioning as an enzyme. Okay, so what are some of the applications that we're ultimately hoping to develop through a structural understanding of catalytic DNA? One of the simplest things is their applications as a diagnostic agent or a biosensor. And these have been developed for a while here, and they generally function as follow. We will have an enzyme strand bound to some sort of substrate that bring a fluorescent fluorophore and a quencher in close proximity. Fluorescence is off. We can then introduce the cofactor, which will allow the DNA enzyme to getting its proper fold, have an activity that would cleave. This cleavage would release the fluorescence probe away from its quencher. We turn on fluorescence and we get a signal. So what are the kind of cofactors that have been used so far and actually brought into the field for these types of biosensors? One of the earliest and most promising was sensing mercury. I also am very passionate about this because West Virginia, where I live these days, has a rich mining history, as you may know of, and there is a lot of contamination of heavy metals from that history and that past. So how would we sense mercury through DNA? Well, one of the really cool things about this particular metal is it can actually sit between thymines and cause a metal-mediated base pair. So in the absence of mercury, we have a rather flexible, rather open thymine junction. When the mercury is present, it would zipper it together, allow this to get to a conformation, which activates the cleavage, turns on our fluorescence. If anybody has questions while I'm going, please interrupt me. I'm happy to do them on the fly, okay? So the other reason that I'm really excited about catalytic DNAs is their potential applications as therapeutic knockdowns. Okay, so we know that a lot of diseases are caused by mutations that lead to non-functional or misfunctional proteins. And what I'm trying to represent here is we're transcribing, say, 2 different RNAs. One is wild type, goes off to make a normal protein. However, if we have a single nucleotide mutation, when we export and translate, maybe we get a non-functional protein. Maybe that's a dominant negative non-functional protein. It's causing disease. And the idea is that we could introduce these small oligonucleotide DNAs that function as enzymes, exploiting the unique binding arms that they encode. We could recognize and cleave with unique specificity that mutated transcript, leave the wild type transcript in place and functional, and hopefully we would be able to maybe relieve the effects of disease, if not cure it, if we hit an early enough therapeutic window. One of the other advantages of a DNA enzyme is that these are standalone machines. So if we think about equivalent machinery, say with morpholinos, microRNAs, CRISPR-Cas9, these are all therapeutic options which either require a large array of proteins to do a biogenesis or to have assembly of multiple components which you would have to get into the cell together. It really makes it much more complicated compared to a single standalone catalytically active DNA. Okay, so all that said, if they're so wonderful, how come we don't see them in the clinic all the time? So we're faced with a critical knowledge gap currently. So we can evolve many different catalytic DNAs by this in vitro evolution in a test tube, and they perform really great in vitro. The only problem is the current conditions that we get this enzymatic activity in a test tube are very far from optimal for supporting any of this activity within a cell. So if we can't get to a more robust active enzyme within a living system, we can't really sustain the kind of turnover knockdown kinetics that we're ultimately hoping to achieve with this as a therapeutic. So one of the critical barriers to this is, until very recently, we've had virtually no structural information about how these DNAs are folding into interesting shapes or promoting any of these reactions. So while you can brute force make oligonucleotide modifications and try to put them into cells and hope for the best, right now the technology is like looking for a needle in a haystack in the dark. So this is the gap we're trying to address. So I knew when I started my own lab, about 5-6 years ago, I really wanted to move into this space. And one of the big reasons is quick scan of a database of DNA enzymes. There are literally hundreds, almost nearing on 1,000 now, different examples of DNA enzymes, and most of these have very unique catalytic properties. So it's a tremendously diverse and rich space for a young investigator to get into. And it was only, I think, 1 year after I set up my lab that the first DNA structure came out showing how you can get ligation through a structured DNA. As we were doing the study I'm going to tell you about today, one of these RNA cleaving enzymes came out, as well as a rather controversial but very useful NMR structure of the same thing that we're studying. So this was the state of the field at the time of publication. Okay, so in 30 years since evolving the first DNA enzyme, I am hardly the first person to come up with the idea to try to crystallize these. What was the state of the field when we came in and started trying this? Well, there were a couple different crystal structures out there, but they were all concluded to be non-informative. So we would take our DNA substrate strand, our RNA substrate strand, and our DNA enzyme and try to anneal them together. And over again, what came out of the crystallization were these holiday junction complexes, where instead of forming a one-to-one substrate to enzyme dimer, we were actually getting large assemblies, and they were winning the day in crystallization. The primary cause of this turns out to be a palindromic sequence within the core of the in vitro evolved sequence of the catalytic DNA. And unfortunately, it's shown here in red as a site that is practically invariant. You cannot do mutations to these or you lose your activity. So unfortunately, the thing driving crystallization artifacts, we are enzymatically married to, and we need to keep it in there moving forward. So how are we going to change this? How can we avoid these pitfalls? Well, we reason that if we put everything on one single chain, that maybe we could largely mitigate the effects of these annealing artifacts. So what we designed here was to have our DNA enzyme catalytic core out here, two different flanking arms. The arrow is where we have our purine-pyrimidine junction. This is where the cleavage is going to occur. And we capped off with a GAAA stem loop. Importantly, we need to verify that the thing we're trying to crystallize is actually functional. So we put a fluorophore on one end, incubated with some magnesium, and you'd see cleavage. Okay, cool. A one-strand kind of situation can actually work. But this is going to be sitting around in a crystal tray for days, weeks, if not months for initial screening. So we need to have a way to keep things pre-catalytic. How can we accomplish that? So we got some inspiration looking at backbone cleavage and the presumed mechanism of action of these DNA enzymes. And what people think happens is through an unknown base, we get activation of a 2' attacking nucleophile, and this is at this conserved purine here. This attacks the sissyl phosphate and causes the break in the backbone, resulting in a cyclic 2', 3' kind of arrangement here. So we thought, that's great. Can we just block the nucleophile? Okay, so we made a 2' O-methyl substitution, and in subsequent biochemistry, we could show that we had blocked the cleavage, but we were confident that we still had things folding into a catalytically relevant structure. Okay, so next we tried to crystallize, and I'll briefly describe our efforts here. We tried a lot of different things. One of the first things we tried was actually not what's shown. What we tried first, we had this GAAA, we actually introduced this as a kissing loop that we see in some viruses. And the idea is that these loops would come out and base pair together and hybridize. Screened about 50 constructs with that idea, got nothing, or the crystals we got just would not diffract. We also tried a strategy where we extended a single stranded region here that would promote annealing at that position. And we tested a number of different constructs and sequence lengths. That was not productive either. We got really excited though when we had this GAAA loop. And what's thought to happen with these is that they can reach out and form random kind of insertions into neighboring helices to organize, nucleate, and promote your crystallization. The crystals looked aesthetically just beautiful. However, we were stuck with 8 angstrom resolution. And we really poured a lot of effort into optimizing this rotation. I was just talking with Wilma that another strategy we used was the length that gave us crystals. We would try to introduce non-canonical base pairs in here, which would cause just a little shift. And that's been very productive in some RNA crystallography, but we are really against a wall with this DNA enzyme. Fortunately, about that same time, the 817 structure was published, and they had taken a completely different method to drive their crystallization. So what this group had done is they had taken a fragment from an avian swine flu virus, Pol-10, which binds to blunt double-stranded DNA ends. Awesome. We have one of those in our structure already. So when Evan Kramer joined the lab, this structure had just come out. First week, express this protein, cleave it, get it into trays. We did still have to screen roughly 10 to 15 different lengths of things, but we did find crystals that looked like this. Not as aesthetically pleasing, but here's 2 hot tips for anybody who wants to be a budding crystallographer. Don't get caught up on looks, okay? Sometimes these ugly crystals are your winners. The other thing when you have these rods is to take a moment and scan throughout the rod and see if there is a sweet spot in them. Sometimes within these long rod crystals, the ends diffract better in the middle or vice versa. So that ended up being really important to find a place that we got 2.5 angstroms. The other hot tip, and this was a trick I learned from Jamie Kate, is, believe it or not, how you freeze the crystals made a big difference. Plunging these rods into liquid nitrogen this way, no diffraction. Plunging it this way, great diffraction. So try even some different angles when you're prepping your crystals, and try not to lose hope. Okay. So with that diffraction in hand, another great thing about this project is we had this protein to allow phasing. This is what the original phasing maps look like. Building the model, the best strategy was actually to build backbone into this and then refine. One of the excellent things about this phasing strategy too was, hey, we know that this protein binds to blunt ends. That is critical information when you're trying to do structures of nucleic acids. You only have a 4-letter alphabet, and it is tremendously hard to know where you are in your sequence sometimes, which makes your model building rather difficult. Okay, so we ended up with a structure with basically 2 DNA enzymes annealed together and a substrate strand coming between. So despite our best efforts, we also fell into a dimer trap. However, unlike the other crystal structures, we saw interesting architecture surrounding the point that the sissyl phosphate is located. We tried to prove if this dimer has any function. So what we did is we first looked at this palindromic sequence that is mediating the dimer formation and asked, can this form in solution? And luckily, this site is actually the palindromic cut site of NHE1. So if we took the DNA enzyme and just incubated it in long incubations with this restriction enzyme, indeed, we could see that we were getting digestion, showing that the DNA is dimerizing, making this site for enzyme digestion. However, I think everybody will agree with me that this is not a major population in solution. We, of course, also tried this by gel shift assays, and through a number of different follow-up experiments I'm not going to show today, 5% in solution was a generous estimate. So sometimes crystallization will give you what crystallizes best, not what is most likely to happen in solution. Okay, but why were we still excited about this? So when we looked at the catalytic core arrangement versus where the sissyl phosphate, the place action is happening, we actually saw 2 very interesting things. One is that there was a very sharp distortion of the backbone immediately before where we do all our clipping, okay? These kind of sharp turns and reversals are highly, highly suggestive of properly folded, mechanistically informative structures. We see these kind of backbone reversals in things like self-cleaving ribozymes, and we were delighted to see that mimicked in this structure. We also saw an arrangement of 3 magnesium ions coordinated to the scissile phosphate where we're doing all the cleavage. And the coordination pattern of this actually made a lot of sense for what we would expect to see for activation of a nucleophile, relieving the pressures of a reaction state intermediate. But there was one big problem here, is that we actually didn't have the proper geometry to really nail down what we were seeing as a functional active site. So normally when we think about a 2' hydroxyl attacking and cleaving a phosphate, there's something known as an inline attack. And the coordination geometry that we captured did not support this. We could do some modeling, and we predicted that if we did basically a 180-degree flip of the purine in here, we would be able to obtain this highly characteristic inline attack that would support the RNA cleavage. That's okay. This might actually be a clue that suggests that these DNA enzymes require high amounts of magnesium to really facilitate and lock in this conformational change. It's the kind of information we really wanted to find, if you think about it, when we set out to get a structure. Because what's limiting these things to be able to function under physiological conditions? This is the sort of information that suggests ways that we could optimize these things. Okay, so we get to the end of the study. We've got a lot of kind of interesting things, but we kind of had the jam taken out of our donut a little bit, right? We didn't get a monomer structure, and we're left with this situation where I don't know if we could be more clever to prevent getting dimers after dimers by crystallization. These palindromic sequences that are forming this dimer interface are such a powerful force of crystallization, we are never going to get away from it. So what else are we left with? Well, we could do SACS. SACS doesn't care about size, but it's too low of resolution to really be of use, in our minds at least, to give us clues on how to make these enzymes more functional. There's an NMR structure out there already. NMR is not really my specialty, so I don't necessarily want to play in that pool. But over and over again, we talk to people and they're like, how about cryo-EM? Well, yes, there's a cryo-EM revolution going on, but you still have to be big enough to be seen even by an electron microscope. And 15 oligonucleotide DNAs are not at that threshold, okay? So recent efforts I'm going to show you now, and I'm looking for feedback on this stuff, and I'd love your insights, are the directions that we're taking to get away from crystallography of DNA enzymes and try to exploit cryo-EM as a way that we could rapidly get new structures of catalytic DNAs. I'm particularly interested in this too, because I'm sick of trying for years of different rotations to get to something that diffracts. I would love to just have a pipeline that we could study these things rapidly. Okay, so the first thing we thought is maybe we can take advantage of DNA nanostructures or DNA origami. And I remember as a PhD student, when this Nature paper first came out, we were taking these long, I don't know why that pointer's not coming, scaffold DNAs. So several kilobases of DNA that you make by M13 rolling circle replication. You then put in these very specific staples, and the staples will go in and organize that long scaffold into an astonishing array of shapes. I think the first paper had something like a smiley face in it, and I was always just like, that's so cool. But is it useful, right? So it was satisfying to come around and think, well, maybe we could take these large nanostructure scaffolds and have it be something that we could embed a DNA enzyme in, or embed the substrate that we're trying to cleave, get us over the size threshold that we could visualize these things. Heck, maybe we could do several at once. We could put like one DNA enzyme in one eye of the smiley face and another one in the other. So we started by designing these non-symmetrical large scaffolds because we want to have a way to orient these when we do single particle analysis. So we introduced some different notches at the ends. And then in the middle, we had an aperture that we basically embedded the 1023 DNazyme in, and then asked, can we put a substrate in? Can we get activity from a DNazyme embedded in a large nanostructure like this? So we could, or I wouldn't be showing it to you. Okay, we could see that the substrate would come in and shift by FAM, and then we could also see that it was cleaving. Looks good. Great. Let's get to the microscope. So in negative stain, it looked highly promising. Okay. So we had these lovely little discs that my students insist look like Cheez-Its. And more excitingly, we could actually see some different orientations. I was very, very concerned that all we'd end up with is a mosaic tile, and we would never get different orientations that we could do any reconstruction from. So seeing this kind of arrangement in negative stain gave us a lot of confidence to go forward and get into ice. And as fortunately, when you go into ice, it's usually where the wheels come off the bus in cryo. And we actually had the opposite problem. All my Cheez-Its turned into, I guess, rotini. So everything was sitting in one orientation, almost like a quarter sitting on its edge. And we collected 10,000 images, and I think we came up with under 100 particles that were in any different orientation than that. So we tried detergent screens to get over this. It ultimately was just really non-productive, but I'm open to suggestions on things to try, different strategies to move this forward, because it was very, very promising. So the other thing we've been trying is, you pretty much calibrate your microscopes these days with a ribosome. How about a ribosome as a display system? So we got a little lucky in that E. coli has one subunit of its large subunit called L9, which is dispensable. Okay, it's not needed. The cells can live. So there was a CRISPR strain that had L9 deleted, and we reasoned that maybe we could fuse DNA binding proteins on the L9, use this as a way to pull assembled ribosomes out, put our DNazyme on, and potentially visualize through the ribosome. So step one was to try to find candidate DNA binding proteins. So we scanned the PDB and we were just hunting for things that had a very strong N-terminal alpha helice. These were some of the candidates we came up with. You gotta love the PDB, okay? This really helped a lot. We've obviously tried a lot. I'm just gonna show you the one we got data for last week, which was a fusion with EcoR5. So we have a version of this that's mutated so it can bind DNA but not cleave it. AlphaFold 2 has really been essential here too, okay? So we would take our L9 protein in our proposed fusion and we'd play with the rotations, okay? To try to predict an orientation that would get the assemblies that we want, but not sterically block or hinder the assembly of DNA into these large complexes. So this is kind of what it looks like. We have our AlphaFold kind of part of the pipeline, and then we'll take pre-existing ribosome structures, do a little symmetry dance, and ask, does this look reasonable to support binding activity? Next, we had to purify the ribosomes. So we have our L9 protein with a his tag. We go into these strains that have the endogenous L9 knocked out. We can pull out the ribosomes. We can see the different subunits coming out. If we change the salt conditions, we can actually enrich for a 50S versus a 70S. So we have a lot of nice flexibility. We can see beautiful images by negative stain TEM, but more importantly, the EcoR5 that was fused onto L9 was active for shifts in gel shift assays at a molar ratio that is reasonable. I think we lost maybe twofold activity of affinity through the fusion. An interesting thing when we are in negative stain, though, is that if we didn't add DNA to the sample, so I'm kind of zoomed out, all these little spots here are 50S ribosomes, we would see a couple of them dimerized like we would expect, because restriction enzymes are obligate dimers, okay? We didn't see that unless we put the DNA in, and the DNA would zipper together these different ribosomes. So I suspect that we don't actually have the best orientation of our fusion yet, and we're going to have to put some more work in at this point. But more excitingly, when we went into ice, unlike with our nanostructures, the ice was fantastic, okay? We could see dimerized ribosomes sitting next to each other. It's a little frustrating. We don't have a little more time to play with it. We can actually see things sitting between the ribosomes. So I do feel like we're getting our DNA enzymes in there. One of the things that we're fighting against right now is that the software that does reconstruction, okay, it is used to centering on a large body and doing 2D classifications in that manner. What we're going to require out of this is that we center at a fusion point between two large different lobes. And I'm having a real big time right now convincing CryoSpark that that's something that we really want to do. So we might need to kind of get under the hood and change the parameters a bit. But I'm very hopeful that this might be a strategy that we can rapidly get structures of DNA enzymes in the near future. Okay, other developments recently. So we're also exploring new mass spec methods to support this research. And what I'm showing here is a new type of ionization that we have a very well-funded score, hopefully going to get our letter of award soon. So the problem with mass spec for nucleic acids is you need to run the machine in negative ion mode, which has terrible, terrible sensitivity. So if you want to do mass spec of nucleic acids, we need a way to boost the sensitivity several orders of magnitude to allow us to do native mass spec to try to do some studies to understand what's exposed to the solvent, what's dynamic. And what you're seeing here is a little power source attached onto a sharp capillary. In this case, it's a cover slip. The inlet tube is mounted on this. And when you introduce an electrical current, it vibrates very rapidly, the sharp edge vibration, which causes your nucleic acids to be ionized in this little plume of smoke here, which is then sucked into a capillary and sent off for time of flight. What we saw using this method of ionization is that we were modestly getting 100-fold increase in sensitivity run in negative ion mode. So all of a sudden, in solution, dynamic mass spec experiments are totally on the table. Proof of concept of this was doing quadruplexes, okay? So DNAs that can zipper together in these quartets and form structures. So what we're doing is we're taking one side native and one side that's exchanged with hydrogen deuterium exchange. The idea is that if you're open, you'll exchange for the heavier isotope of deuterium. If you're closed, less flexible, more kind of on the inside, that exchange doesn't happen as rapidly. And in the quadruplex structures, indeed, we could see that this was happening, and that was our proof of concept to move forward. Okay, in the last couple minutes, I want to just kind of talk about the future aspirations of these DNA enzymes as therapeutics, okay? So I'm at the age now where I think less about the academic thrill of discovering something just for the sake of it. And you get to a point you really want to make an impact. And working in a medical school in West Virginia, we see a lot of diseases very indicative to that area. You also have to lean into the strengths of where you are. So half of my department are vision researchers. We have a very large contingent of people. So how can we actually take these things from the bench to the bedside? And in West Virginia, there was a lot of vision disorders, primarily associated with this disease called retinitis pigmentosa. And this is caused by a mutation in rhodopsin. This is what binds to the light sensing kind of floor that makes your eyes function. One mutation is giving this dominant negative effect, which is the perfect case for how you could actually use a DNA enzyme as a therapeutic. The other great thing about the eyes, they're not only the windows to the soul and the brain, they're also incredibly accessible if you want to try to get things into them. So very nice solution. And this is what this disease looks like. Over time, your vision basically degrades. You get this tunneling effect. It causes a tremendous loss of quality of life. So even if we don't cure a disease, if we can mitigate a disease and catch it early, the quality of life improvement could be dramatic. The other thing we're thinking about targeting going forward are myosins, or just kind of any large protein family with high structure conservation, but low sequence conservation. So through a recent faculty hire, Rong Lu, I've really come to appreciate that myosins are a very, very large family, and some of them are highly associated with particular diseases. So there's specific myosins with mutations that are associated with deafness. The only problem is trying to target this highly conserved protein structure leads to a tremendous amount of off-target effects, which are just a nightmare to sort through. But because myosins have such low sequence conservation, DNA enzymes could potentially be a perfect application treatment for these kind of diseases associated with hearing degeneration. Once again, the ear is very accessible. We can get things into cells in the ear a lot easier than we could try to get something in the brain. So currently, we got to get these things working better in cells. And we've had a raging debate on, do we try to go for a fluorescent assay for this? Do we try to go for a live dead assay? We're just getting the system together. We're starting off with fluorescence, where we're hoping to have a two-color system. We introduced the DNA enzymes guided and improved by our emerging structures, have a way to monitor the increased efficiency within cells. That is still the critical hurdle to get from the bench to the bedside. So we're developing the structural stuff that I showed you just now, but we also really need this cellular arm for my aspirations to actually become a reality. Okay, that pretty much brings me right at time. I want to acknowledge the people who did this work. Believe it or not, this work is primarily all done by Evan Kramer in the lab. So Evan has been the students I challenged to really spearhead this new direction of my lab. Sarah does formation of large splicing ribozymes. Ryland studies how these things move and invade different parts of genomes. I'd be happy to talk to anybody individually about that. Talented technicians, Steve Valentine, who does a lot of our mass spec collaborations. And I got to give a huge shout out to our collaborators at NECAT in the Advanced Photon Source, who worked with us tirelessly throughout the night to study the tremendous amount of crystals for the first project that I showed you. Thank you so much for the invitation. It's been so much fun visiting. Thank you.
We have time for questions.
Great, thank you. Thank you for the talk. One brief question. How, you mentioned you had 5% of that DNA enzyme dimer in solution. Did you look at a concentration dependence to see if you added more, if you had a percentage of dimer shift up?
We did try those studies and we couldn't find evidence that there was a threshold that we could really drive it. We also debated going into single molecule studies to play with this in a flow cell, but we just weren't getting promising enough concentration kinetics that seemed like it was going to be a very fruitful path to go down. But it's a great question. One thing we did try to sell when the paper was in review is maybe this dimerization is transient by necessity, that this dimerization promotes some sort of shape that makes the chemistry happen and then it releases. I still don't mind that hypothesis, but we'd have to put a lot more effort into trying to prove it.
I'm just wondering, can you incorporate these DNA sequences into a single strand phage or virus as a delivery system?
Yes, I think we could. That's another reason we love the eye system so much. We have a faculty member who does adenovirus infection for eyes to treat a different type of eye disease that's specifically like the blue cones. And she has very promising work that the viruses are delivering her particular gene therapy solution. And hopefully, I mean, that would just be tremendous if we get some editing and fixing of that.
So may I ask two questions?
Yes.
Oh, you want to go for it?
Sure, I was just gonna read a question.
Oh, from the chat, please.
No problem. So there was a question from Arthur online. What if you use a tetrahedron or other polyhedron DNA or origami structure with 1023 in the center of only one face? It should eliminate a preferential orientation issue, and aligning such particles should be pretty straightforward as well, since you have 1023 anchor on only one face.
So, yeah. So that's a tremendous idea. And I'd really be excited to try different shapes of nanostructures. You know, to be honest, we didn't explore too many of them. I think just because the ribosome display stuff started to look very promising. But it is something we want to follow up on. And I love the tetrahedron and the non-symmetry idea, because I would hate also to end up in a situation where the details that we're getting are trying to be smoothed out by the symmetry alignment, by the nature of how you align the particles. So it's a great suggestion.
Thank you, Arthur.
Okay, we have one more question here.
Hi, I just had a question on how like the spray ionization native mass spectrometry, like, could you go into a little more detail how it helped you?
On how that'll help?
How that helped you, like helped you,
Oh, so how did it help you, or like? Oh, so this is something that we're just starting to go. We haven't actually put the DNAzyme through its paces yet for this. We just did the quadruplex structure as a proof of principle. So what we envision is that the inherent flexibility of the 1023 DNAzyme is what's potentially limiting its effectiveness from being locked down and active under more physiological conditions. So if we have a way to monitor flexibility, in solution, real time by HDX exchange, we could take random pools and actually get to potential very interesting avenues of optimization in a much more systematic and speedy approach. Ultimately, this is another tool. I don't think one tool is going to get us there. I think that in my mind, we're going to be hybrid. Okay, we're going to have hopefully a structural cryo-EM arm going together with mass spec to support what it looks like dynamic in solution. That could be also supported by small angle X-ray scattering, for example, and ultimately getting the robust cell assay to really get it in and show that it still functions in a cellular context. So the best way I think that we're going to get to a good solution is to have all these different arms working and communicating and informing us, supporting their efforts. That's a great question, though.
Yeah, thank you. So I got a few questions, if I may. Okay, thanks. The first was the 1023. How good an enzyme is that in the optimal conditions in vitro?
Optimal conditions in vitro, it can be very fast, but it requires above 25 millimolar magnesium. which is a far cry from 1
from
to 2
what's in the
millimole.
cell. Yeah,
Yeah.
yeah, fair enough. Why are there so many people wearing Canadian flags in the photo?
So this is from, it turns out I'm a Halloween costume this year. So I showed up on Halloween and I'm like, man, that's a lot of good looking flannel you all have on.
I was gonna ask about that.
And then we showed up to group meeting and they all had Canadian flags on. I was like, oh gosh, I'm the costume
Okay,
here.
all right. Yeah, well, it's important to maintain a sense of humility and humor
It's
in the
what-
face of things like that. In terms of trying to get so I think the idea of going into the eye where you can do a direct injection for RP is terrific. I mean, that's standard of care now for people with macular degeneration, where you're injecting antibodies. So what about using something like a PNA to improve the likelihood of cell penetration?
Yeah, that's a that's a wonderful point, is delivery is still going to be something that we really need to optimize. You know,
Right.
so PNA can be very useful for this if we don't go the adeno route. You know, why don't we have DNA vaccines? Well, it turns out that people don't love being electroshocked. So to get it in, that was
Yeah,
like the limiting thing.
Yeah,
So,
right.
you know,
Yeah.
that's the other, I guess that's another arm we have to think about, is not only optimizing, but really thinking ahead for what an effective delivery ultimately
Right.
will be.
So I think the good news there is that between things like PNAs and the advances that have been made for in the CNS realm for injecting RNAi into into the spinal column and getting effects in the nervous system. One should be able to understand some of the lessons from the changes to the backbone that were implemented there
Yeah,
to try to improve cell penetration.
I 100% agree, and I'm
Yeah.
glad that we're not starting from for once, a completely black
Yeah.
block. So
Absolutely. Yeah, absolutely. Do you want anything more?
Are
Anything more
there any
online?
other questions?
Anything more in the room? We've got nearly as many people online as we have in the room, so that's great. That's the value of having the hybrid format. And so before we close, let me just give you the couple of pieces of PDB swag that I promised you earlier. So the first, which is of transient value, is a 2024 calendar assembled by David Goodsell, and of truly enduring value is a RCSB PDB Yeti that will last longer than you if you take care of it. So thanks a lot. Great talk.
Thank you so much.
Okay.